People don't know where research topics come from. And the cool thing about science is that, or academic science, is that I get to have an idea, and if I can get somebody to give me money for it, or if I can do it for free on my own time, or if I can talk a student into doing it, then we get to do what we want to do, right? And my boss is not telling me what I have to study. I get a lot of questions about like, oh, do you are you going to be studying different things when you move jobs or something like that? Like, no, I'm going to continue to study whatever the heck I want to study.
Yeah.
You're on a ship, either have good days or bad days. A bad day is defined as it's nighttime, you've lost power, you're in heavy seas, and something's on fire, okay? So it's really nice because most of your days on a ship are really good days. But if we're having a bad day, that's what you have to be prepared to self-rescue from.
Hi, I'm Nina, a filmmaker on a mission to uncover the hidden stories of science. In this series, I'll bring you behind the scenes aboard the NSF research vessel Sikuliaq, where I tagged along with an ambitious group of scientists, technicians, and crew members on a 2-week expedition in the Gulf of Alaska. We delved into the ocean's depths, and through sediment coring, we're not just pulling up mud. We're uncovering the secrets of life on our planet, revealing the intricate cycles that shape our environment, and understanding why this research is crucial for our future. Get ready to come along for the ride.
My name's Drew Steen. I'm an associate professor in the Department of Biology at the University of Southern California as of yesterday. Was before that at the University of Tennessee. We have two chief scientists on this cruise because we have 2 NSF grants that have funded the cruise. Mine is to better understand the deep subsurface biosphere. So if we go out virtually anywhere in the ocean and we go to the bottom of the ocean and we put a corer or a drill or whatever kind of device that we have, we can keep going into the earth for, feet and possibly miles, possibly many miles, until as long as the temperature stays below about 60 or 80 Celsius. So that's up to about like maybe 140, 160 Fahrenheit, something like that. Anytime the temperature is cooler than that, basically anywhere on planet Earth, you will find microorganisms that are alive. And these microorganisms that live in the deep biosphere, so deep rocks in the Earth's underneath the continents or deep marine sediments, are really mysterious. They live on almost no energy. In many cases, they've been isolated from the surface world for thousands or even millions of years. And they live this really bizarre slow lifestyle. So we think that they sort of turn their carbon over in their bodies. This is a little bit like a microbial lifespan of like 1,000 years. Some people have even said 10,000 years. It's really kind of blows my mind to imagine a single cell being alive for 1,000 years. If we think about cells that we know a lot more about that we can grow easily in lab, they might divide every 20 minutes. A really slow growing cell might divide every day or 2 days or 10 days. That would be exceptionally slow. And these cells are dividing, less often than the entire lifespan of a person, which blows my mind. And biologically, we don't understand how they do that. And so fundamentally, what I'm trying to do is understand what the adaptations that these organisms have are to living very slowly. And then can we use some of those adaptations to improve human processes, to be more useful in industrial processes, or to help us deal with situations where we would like cells to sort of stay at a maintenance state for a long time.
Mountain view.
Yeah, exactly. So I'm not really trying to get people to live forever, but I am maybe interested in figuring out how to get organs that are being transplanted to, be able to stay outside of a body for a longer time or something like that. Or maybe more realistically, how to develop microbes that produce useful products for humans in a way that uses less energy and is more efficient.
And we'll be able to tell you what microbial species were present in the mud that we're looking at, roughly what kinds of genes the most abundant species have, and so therefore what kinds of things they're doing to the environment. And then the big goal then is to understand a little bit about how those genes tell us how those organisms are adapted to life buried deeply under sediment, where they're out of contact from the surface for many thousands of years.
So that's one of our projects. Allie can tell you a little bit more about her project.
Okay, so the work that I was trying to do on this trip, which represents about half of the work that I do as my research, is thinking about how the seafloor controls how chemicals move between the mud that exists below it and the water that exists above it. And so sometimes when we think of the seafloor, we think of kind of a boring place, we think of mud, but it's actually a really active habitat. There's a lot happening there. We can see things like crabs. We can see these bubbles that represent methane that's being released by microbes. So we have this really active community where critters from teeny tiny things like what Drew and his group are studying, all the way up to much bigger things like some of the things we pulled up in our cores, are impacting the environment by eating in the mud, digging around in the mud, pooping in the mud, and that really drives changes in the ocean. So what can happen here will control how carbon and nutrients and trace metals are either trapped and removed from the ocean or released back into the water. And so this can control things like how quickly climate change happens, how big of an ecosystem the ocean can support, if there are toxic things like mercury in the water that your fish is eating. So the question that we were testing on this trip was, how is iron being stored and mobilized from Gulf of Alaska marginal settings? But within the Gulf of Alaska, trying to understand the role that sediments play in trapping or releasing iron, which again drives all of those other ocean chemical changes. The first place that I really started to ask these questions was within the Arctic. The Arctic Ocean's a really weird ocean because it has a lot of these really shallow areas that are represented by the light blue colors. Those are places where the seafloor is really close to the surface. And so there's a lot more of those shallow areas than in other oceans, which make it a really good place to ask these questions about the links between the mud at the bottom of the ocean and the water that sits over it. The reason that I was so interested in this area for a long time was because it's a place where there's really rapid sea ice retreat. So we're causing the whole ecosystem to really experience this really rapid change that kind of creates a natural laboratory to see what happens when you fundamentally reorganize a surface ecosystem, which impacts the seafloor. On my first expedition, I got really, really excited about iron. So we were doing trawls and we started pulling up these like huge fist-sized chunks of iron oxide minerals, precipitating basically everywhere, which highlighted that it was a really kind of unique iron cycle that we were seeing in these locations. Iron operates in a really weird way in places that have glaciers. So you can think of glaciers almost as a bulldozer. They move over the landscape, powder up the rock, release a bunch of iron, and then eventually it precipitates or actually just rusts on the landscape. In the last few months, you might have seen some headlines of similar things here in Alaska, that scientists are starting to notice that a lot of Alaskan rivers are turning orange. I think it's 75 rivers within the state have satellite images that show that they're basically turning rusty. This is a similar process. What we're seeing is the melting of permafrost throughout the state, releasing a bunch of iron to waters like rivers that then flow out into the ocean. And so the iron cycle within systems like places where there are glaciers or places where there's permafrost are changing really rapidly in a way that we can see kind of year to year. We care about that because iron, as well as manganese minerals, do a really good job of trapping chemicals within their kind of solid phases. So what the iron's doing will impact, again, things like mercury and arsenic, which we care about because they're toxic, nutrients, ecosystems. We also tend to care about iron because it can be a biolimiting nutrient. What that means is that process of photosynthesis is actually controlled by how much iron is in the water column. It is the biolimiting nutrient where we are right now. Algae that are photosynthesizing in the surface, they take CO2, they turn it into organic material, and that organic material can make it down to the seafloor. And so what we're really trying to understand is how the efficiency of that process of producing organic material and removing it out of the ocean, how that'll change as sea ice retreats. Iron limits productivity in the Northern Pacific, and a lot of the iron that actually makes it out into the Northern Pacific comes from sediment. So it's being remobilized and transported from the kinds of places that we've been coring over the last 2 weeks. So that let us set up this really simple hypothesis that we're increasing atmospheric CO2. That causes the Earth to warm. Warming Earth makes the sea ice retreat. But now that you have these big shelf seas, maybe that means that there's more time and space for algae to take that CO2, photosynthesize, and create organic material. That causes a loss of carbon from the system, which can maybe let carbon be trapped in the seafloor and slow climate change. Oh, I didn't know there were questions. Excellent. Yes, Kendall.
How are you anticipating that percolating down the food chain? Like, are you anticipating more blooms in the Gulf?
So I think it will depend. So to get into the nitty-gritty details, the Yakutat site is actually probably the site that is right now the most efficient at iron transport. Tidewater glaciers are great because the glacier goes out into the water, so it provides a direct pathway for the iron to make it out before it precipitates as rusty iron oxides. So I would actually expect that as we lose more and more of those tidewater glaciers, you're likely to actually lose the iron sourced from land. So then the question is, to what extent do the sediments themselves hold on to it and release it slowly through time? And I'll let you know in a year or 5.
There's no wind over here if you guys notice. Yeah, thank you. All right.
You are all the scientists, the core labeling technicians, amongst other things. The core tubes are your sample tubes. They are your sample vials, they are your sample bags. It's your science or your boss's science that depends on it, so you are the stakeholders in seeing that the labeling is done correctly. A core without good labeling is totally worthless, right? As worthless as any other sample that is not connected to its metadata. So we just need to make sure we do it right, and also consistently, and also according to the format of the core repository where these will go live on for all the other scientists in the future. Every core will start off with the same prefix, which is ship, SKQ. Since these cores will be going to the Oregon State University Marine Geological Repository, their convention is year and month that the crews start on. So this will be SKQ 2407. That's not a good set. You will label your cores right side up, most likely, instead of upside down. Number of deployment. So the first one will be 1. If that first one shall mayhaps be a multi-core, once my spidey sense tells me it might be, then it would be 1 MC. The gravity core. A piston core, it would be 3 JC. The J is for jumbo. There will also be a 3 TC for the trigger core, because that littler gravity core is the triggering weight that triggers the free fall of the piston core. The gravity cores, piston cores, and the trigger cores, they're all going to be in that white PVC, and they all potentially will be more than one section in length.
What's your standard section?
Standard section length is 150 centimeters. So the order of operations, which we'll do is in smaller groups, is you draw a big straight line down it, then you go and measure it, and put the circles on, and then you do as much pre-labeling as you can.
You really said precision.
Say go fast. I'm going fast.
No, this is great. All right. You hold that measuring tape at the base of the threads.
I didn't see over there. How close do I stand?
A little bit further.
Hello, I'm Chris Fancher, coring technician.
I'm Ben Freiberg, and I'm also a coring tech. And what's your guys' role on the ship?
Put the mud in the tube.
That's really it.
Yeah, yeah, we do the part up until they start doing science on it, basically. Bring the things, put them together, throw them over the side, bring them up, tubes mostly, and then hopefully there's mud in them, at which point the scientists can do whatever it is they're doing in there. And now let's make it 3 people. Pull that out slow, nice and slow. Yep. Okay, now we can work it on here. Yep, so now I got it sold. All right. So Usually they're not so crazy full when you can take it down, but we only have this much water. So now is the time you can pop the lid off, and we know the mud's not gonna fall out.
So you're at 4.4 here?
I don't have a printer. Do we need a crimper?
Methane.
Yeah, so we need to crimp it right away. Okay, so the crimper is over here.
Oh, I'll print it.
Yep, and then shake it up real good. So we have the
Here we go. It's coming through.
got it?
It's coming. Ready?
Okay, yep.
Nice.
Okay, now flip that upside
Flip that
down.
upside
Yeah,
down.
Yep. Flip that upside down. We're trying to.
Full spatula.
Yep. There you go. All right. Give us sweet, sweet methane. Or not, who knows? We have to bring out the napkins. It is very,
keep this little tab with it.
yeah. Okay, perfect. Yeah, we might as well just put it in.
Just try to don't rush the scrape. Yeah. Like that. Perfect. That's awesome.
Alrighty, this is our first sediment sample. We have 2 bags of the sediment, and now we're going to put it into a negative 80-degree freezer. We're just going to pop it right in there. Okay. And now we go repeat it over and over and over again. Goodbye.
Let's just look at what time it is right now.
AM.
And what are you doing?
I'm putting on sunscreen to prepare for the morning so
Because
I
of
don't
the
get my
sun.
ears burned any further.
Yeah,
When
because
it comes up, it comes up
just so it's way too bright outside.
Those UVB rays, man, make me sick.
That's so funny.
I get it, because it's like, when am I going to have the time to put this on when the sun comes up?
Is that like a boat version of honking?
It's a foghorn. Until the fog is gone, we're gonna keep hearing that horn every 2 minutes.
Oh.
Get excited. Oh, we're done. Okay. That was so disorienting.
So what's been going on so far?
So we threw down our first multi-core, and we got a lot of mud. Some say too much mud, so we're gonna do it again and try not to be as good.
Yeah, we're too good. That's my only.
So the tool that I used there for sampling was the same as my favorite tool here, which is in this case, this is a megacorer, which is a lot like our multicorer. What we like the most about them is this little bit of light that you can see through the top. That indicates that we've collected sediment and a really clean water column above it. So we can really understand what's happening at that interface between the mud and the water. We then take these cores. You likely saw some of us doing these kinds of things. We slice some of them to study the mud, but we also vampire some of them with something called a rhizin, so that we can really understand how chemicals are moving between those solid phases and the water that's in our mud.
We stick these things in here called rhizins. They have a little filter on the edge of them, and essentially what it does is we pull a vacuum with a syringe and it pulls the water out at the various depths. So we do like 1 centimeter up to 10 centimeters, and then we go every 2 up to 20, and then after that it's every 5 centimeters. Okay. So you're kind of getting various depths along the core. So this is going to be analyzed for a lot of different geochemical analyses, some of which include carbon, others that include metals like iron. So it's a whole suite of geochemical analysis that we do.
Why are you laughing?
Because you're so cute and you started laughing.
Me trying to remember my name. I'm Megan Winsler, and I am an incoming PhD student at USC. I'm in Drew Steen's lab. I'm feeling good. I think we've gotten our process down, which was really a lot of the battle. The first couple of days, very rough, but
Rough how?
Rough in the way that we did not have our process, like our flow down. So now we have our flow down, like we all know all the different protocols. So I feel much better now. And now I know what things actually are, which is nice. Yeah, the coring, but also the sampling, and then the subsequent things that we're doing on board, like the pore water sampling, I didn't know too much about, and things like that. So
Have things been adjusted as we've gone along?
Oh yeah, things have changed since we even started. Different volumes of things that we're doing, or different containers based on what we have and availability of items. I think our sample sites have changed a little too. So things are always in flux, I feel like, on cruises, which is part of the fun. Yeah. I'm Megan Winsler.
We are coloring styrofoam cups so that we can send them down the bottom of the ocean to see how the pressure changes them, and also have a cool little shot glass. That's a memento.
You're the ambassador for it. There you go. Surprised me that I wasn't as freaked out that we were in the middle of the ocean. I thought I would be more scared that we are in the middle of the ocean.
Now it's just normal, right?
And now it's just like, yep, but I'm in the middle of the ocean, and that's fine. I thought I was gonna be like Hey from Moana. That's what I thought I was gonna be like. That's who I thought I was gonna embody. The chicken.
In this episode, we've explored Drew's research on deep biosphere enzymes, which can survive for over 1,000 years and hold valuable insights into life beneath the ocean floor. We've also covered Allie's research on glacial retreat and its connection to climate change, specifically how the exchange of biolimiting nutrients such as iron between sediment and water impacts algae growth, ecosystems, and the climate. And lastly, we walk through the process of labeling cores, taking samples, storing them, and also testing pore water using rhizomes.
Hey there! Welcome to the RV Sikuliaq. I'm Olivia and this is our room. Come on in.